So in the last video, we had a bit of an overview of how transcriptional regulation is accomplished in eukaryotic cells, and we contrasted that with the more promoter proximal and direct regulation that we see in prokaryotic cells. And I'm sure that it occurred to you to wonder, and you've been thinking about this, and the question that I'm sure that you thought of is, if the pancreatic cell and the red blood cell are expressing different genes because they are expressing different sets of regulatory transcription factors. That's great, but why are they doing that? When did they start doing that? What causes an immature red blood cell to specifically express these transcription factors but not those? And those are very good questions, and that's what this video is going to address. And the answer in general is that these kinds of cell fate specification programs are set in place beginning very, very early in development, no matter what organism you're looking at. We start to see what's called differentiation, which takes an immature cell that doesn't really have a defined cell type and causes it to initiate a genetic program such that it becomes more specialized or more differentiated. And that begins to happen very, very early in development. For example, in humans, our developmental program is 9 months long, but within the first 5 days following fertilization, a lot of the very basic decisions about which cells in this little ball of cells are going to be making up embryo versus extra embryonic tissues like placenta have already been made, and so cells have become restricted in the cell type fates that they can adopt. So what we're going to focus on in this video is the developmental regulation of gene programs and specification of different cell types, and we're going to be looking at this as it plays out in Drosophila melanogaster, the fruit fly, because that is the model organism that was used to really investigate and figure out how this genetic program is specified in embryonic development. And what we're going to be seeing in Drosophila embryogenesis is that, very, very importantly, localization of mRNAs to specific regions of the cell is going to be a really important mechanism for initiating this cell specification process and really kick-starting it. Then what we're going to see is a cascade of regulated transcription factors that start to be expressed in different patterns in different regions of the embryonic body, and that ultimately leads to specification of the embryonic body plan and development of different structures in different regions. All right, so we need to address this question, right? We have a fruit fly, Drosophila melanogaster. It's got a head, it's got a butt. How does it know at this stage of development, this little fertilized egg, how is it supposed to know that it needs to make anterior structures at one region, it needs to make posterior structures at another region, that there has to be a back and a front and a left and a right? How is this cell supposed to know how it's supposed to progress? And what we're going to see is that in Drosophila, as well as other animals, this process of morphogenesis, which is the development of the body shape and structure, is reliant on chemical signals called morphogens. So morphogens are chemical signals that regulate morphogenesis, right? But the way that they do that is by regulating cell differentiation and providing positional information to cells in different regions of the developing embryo in a concentration dependent manner. And what I mean by that is that we would have a specific morphogen, a specific molecule. It could be a protein, could be some other chemical signal, usually it's a protein. And if we have a high concentration of that morphogen, M, then that's going to lead to the specification of cell type A, for example. If there's a low concentration of this morphogen, then that leads to specification of a different cell type, right? So that's what I mean by concentration dependent manner. So if we express this protein and then expose different cells to different concentrations of this protein, we can actually see that initiating different types of signaling pathways or cascades, such that we start to turn on different sets of genes, and ultimately that's going to lead us to specify different cell fates. Now, when does that start to happen? Well, in Drosophila specifically, it really does happen at this stage. Before an egg is even fertilized by a sperm, it has already had established within the egg a axis of polarity along the anterior posterior axis, a dorsal ventral axis that specifies back from front. So even though this egg hasn't even been fertilized yet, it already knows that eventually, once fertilization happens, this part of the egg is going to go on to see the establishment of cells that are going to form anterior structures like the head, like the eye, like the lips, like the antennae. And the posterior region is going to be associated with those other posterior structures, and same for the dorsal ventral axis. So it's back here even before fertilization even takes place. Now, in order to understand the initiating steps of gene regulation that happen in Drosophila, we have to talk a little bit about the development of this egg within a female fly. So we have to know something about how that happens in order to understand this regulation. So what we're looking at here is a follicle. This whole green thing is one follicle within the ovary of a female fly. Now, each follicle contains one egg or oocyte. So here, this large cell outlined in white here, that's the oocyte, that's the egg. But in fruit flies, in addition to that oocyte, there are at least two other cell types that are present that are important for regulating what happens with that oocyte. And we're only going to talk about one of them, which is the nurse cells. So these are the nurse cells over here on the left, right? And they're present within the same follicle. And they regulate and support and nourish this oocyte, right? So that's why they're called nurse cells. These are all cells that mom produced. This cell is a gamete, right? So that's mom's haploid gamete that she's formed. These are just diploid mom cells, normal cells. They produce proteins that the oocyte requires in order to be appropriately regulated and in order to develop appropriately. And these proteins are encoded by genes that are referred to as maternal effect genes, because these cells, these nurse cells, are actually part of mom. They're not part of the gamete, right? They're part of mom's body. And so these nuclei contain the genes that are expressed to regulate development of this oocyte that we call maternal effect genes, because it's mom's genome having an effect on the development of this oocyte once it's fertilized and becomes an embryo. This is the oocyte genome, right? Which once it's fertilized by a sperm, oocyte, is going to become the embryonic genome, right? Once we have fertilization happening, the paternal and the maternal genomes are going to combine within one nucleus, and that genome is then going to be the embryonic genome or the zygotic genome, right? So we combine sperm genome, oocyte genome, and that becomes embryonic as opposed to maternal. Right? So these are cells that are technically part of mom's body. These are the gametes that are going to form the next generation. This genome belongs to the embryo. These cellular genomes belong to mom, if that makes sense. All right. So how does this oocyte, even before it's been fertilized, have that, as we said, anterior to posterior axis already established, back to front axis already established? Well, it's reliant on genes being expressed from these nurse cell nuclei. And we're only going to talk about two genes, but there are actually a whole host of genes that are expressed in this way. We're going to talk about two, and those two are bicoid and nanos. So in this nucleus, the bicoid gene is transcribed. The bicoid mRNA, which is represented as this little blue triangle, is produced, and then it's transported out of this nurse cell into the oocyte and specifically localized to this region of the oocyte. The region of the oocyte that's in direct contact with the nurse cells is where that bicoid mRNA gets localized. A different gene in the genome, the nanos gene, gets expressed within these nurse cell nuclei. That mRNA, which is represented by the little yellow sphere, gets transported into the oocyte likewise, but it gets specifically transported to the opposite end of the oocyte, which is the end that's furthest away from the nurse cells. So just based on the position of these different regions of the oocyte relative to the position of the nurse cells within the follicle, that's how we establish the initial anterior to posterior body axis, because it turns out that this region that was closest to the nurse cells that has the high concentration of bicoid mRNA is going to specify anterior head structures. This region that was furthest from the nurse cells that has the highest concentration of nanos RNA is going to specify posterior structures. All right. Now, why is that going to happen? Because it turns out that these mRNAs, once translated, are going to be specifying their protein products, right? The bicoid protein is specified by the bicoid mRNA. And so at this region of our developing embryo, we're going to have a high concentration of bicoid protein, and at this region of the embryo, we're going to have a high concentration of nanos. And because proteins can diffuse within the cytoplasm, what we're going to see established is what's called a gradient, a concentration gradient, right? These are our morphogens that we were talking about that are going to regulate how these cells in this developing embryo are going to develop, what their specific differentiation program and cell fate specification is going to be based on the concentrations of these morphogens that those cells are going to be exposed to, right? So we take a slightly different view of this. Here's a light microscopy image of a follicle with an unfertilized oocyte. Here's our oocyte. Here are our nurse cells over on the left. Blue is indicating the presence of bicoid mRNA. And so what we can see is that the blue is present here in the nurse cells because that's where the mRNA is being transcribed, remember, from these nurse cell nuclei. It then gets transported into the oocyte. So this is actually inside the oocyte, but then localized and tethered to this specific region of the oocyte. Now, what happens during the maturation of this oocyte is that these nurse cells are eventually going to degenerate, and this oocyte is going to expand to take up the entire space within that follicle sort of cuticle covering that it has. And so a little bit later in development, we go from something that looks like this in the follicle where the oocyte's kind of squished over to the side and we've still got nurse cells, to a state where the oocyte is starting to occupy the entire follicle structure. And so here what we're seeing is the localization of the nanos RNA, mRNA, to what's being specified as the posterior region of the embryo. And this micrograph really shows us very nicely that gradient of protein that begins to be established once that mRNA gets translated, right? The mRNAs are localized here. The protein is synthesized. The protein can then diffuse away from that site where they were created, and we end up with this gradient of protein concentration across the embryo, where the highest concentration is here where the mRNA was localized, and then the concentration decreases as we go away from that. We would see a similar concentration of our bicoid protein that would be high here and then would decrease as we go further from that site of localization as well. All right, so these are our two morphogens. This is how we establish a gradient of protein expression by mRNA localization within the oocyte. But what are these morphogens? What do they do and how do they do it? Well, it turns out that what they are genes that encode regulatory transcription factors. And so these bicoid and nanose proteins are going to be interacting with specific gene sequences within the genome to activate the expression of specific genes, right? So what we're going to see is that depending on where in the embryo a cell is, it's going to be seeing a different concentration of bicoid and nanose, and that's going to regulate which genes begin to be expressed in that cell. All right, so what we have essentially is early in embryonic development in Drosophila, we have an embryo that kind of looks like this, where we have many individual nuclei all present within a common cytoplasm. This is a result of mitotic division without cytokinesis, so that we have one cell cytoplasm with many nuclei within it. And each of these nuclei is being exposed to a different concentration of bicoid and nanose protein. Over here at the posterior end, they're seeing high concentrations of nanose, and over here at the anterior end, they're seeing high concentrations of bicoid, right? And then low concentrations of the other morphogen. So low nanose over here at the anterior, low bicoid over here at the posterior region. And so in each one of these nuclei that's being exposed to these different concentrations of these transcription factors, nanose and bicoid, these nuclei are going to start switching on different specific sets of genes. And so these maternal effect genes, bicoid and nanose, are now going to be starting to activate gene expression programs in the embryonic genome. So remember, we said these are embryonic genes that are now going to start to be expressed within this developing embryo in different patterns based on which genes are getting switched on and off by these different morphogen concentrations. And so these maternal effect genes and their differential expression pattern across the embryo activate different downstream genes, which we're just going to collectively refer to as the segmentation genes. There's several different classes, but the basic idea is very simple. Based on the concentration of these morphogens, transcription factors that these nuclei are seeing, they're going to start to initiate different gene expression programs. And those are going to be maintained as we go from one embryonic development stage to the next. And so initially what we get is just a very gross, blunt specification of anterior to posterior, dorsal to ventral. And then within that specification, we start to get a little bit finer in our specifications that are going on, where we might be seeing specification of anterior structures into head and thoracic versus abdominal regions, right? So we get gene expression programs that are going to be initiated downstream of the maternal effect genes that are now going to control what each and every region of this embryo goes on to develop into being. Every cell at every location along this embryo is seeing a different concentration of different transcription factors. And so every cell essentially is receiving positional information based on those transcription factors that tells it where in the embryo it is, right? And initiates the correct gene expression program for that position in the embryo. Now, as I said, we're just looking at anterior posterior. There's a completely different set of morphogens that controls the dorsal to ventral or back to front gradient. So if we imagine that we are, a cell that ends up being positioned here within the nucleus, we are getting positional information along this AP axis based on the concentrations of bicoid and nanose that we're seeing. We're also getting positional information along the dorsal ventral axis, and as embryogenesis goes on, we're going to be getting positional information about left, right, and other structures that are nearby as well. So initially we establish this body plan, AP, dorsal ventral. We start to specify general regions and then finer regions within those general regions based on the gene expression programs that are being established. Right, so this is kind of what we see along the length of a developing embryo, is that every single position that a cell is at, let's say a cell finds itself in this stripe, is going to be associated with different concentrations and combinations of specific transcriptional regulators, right? And so CRIPL is a protein that's regulated by the presence or absence of nanos. And so we see in that specific yellow stripe that we have these specific concentrations of these transcription factors, and as a result we switch on this specific gene called Eve stripe 2, which defines this specific segment and controls what identity it is going to assume. Now, because what we see in Drosophila is this segmentation or striping pattern that gets established along the AP axis, that's where this name segmentation genes comes from. So these are the embryonic genes that are now being controlled in their expression by the maternal effect genes, those morphogens that had a different protein concentration pattern along the length of the embryo. So as development proceeds and we go further and further along and approach, morphogenesis into the adult, each of those specific segments is going to see different sets of effector genes get switched on finally that are actually going to be responsible for forming the anatomical structures that define that region of the fly. Right at the head, we're going to see development of eye, lips, other mouth parts, antennae, and all of those other structures that are characteristic of the eye, because these specific gene programs that were established here are eventually going to turn on the effector genes that cause these specific structures to form. Likewise, here on the thoracic segments, we're going to see, legs forming on each thoracic segment. On the second thoracic segment, we're going to see the development of wings, and so on. So every segment, once its identity has been established by the segmentation genes, is going to be associated with turning on different effector genes. And these different effector genes are called homeotic genes. This is certainly one large class of effector genes that controls the specification of body structures on these different segments. And these are identified in Drosophila. We now know that they're conserved across phyla. They're really important developmental regulators. But essentially what happens is that once you switch on a homeotic gene that's expressed in a specific region, that's going to be turning on the set of genes that control a particular body structure, eye, antenna, bristle, leg, right, in that region. And so to see how these things work, we can look at what happens when you disrupt their normal expression, all right? So in Drosophila, there is a homeotic gene called ultrabithorax, which is normally expressed in the third thoracic segment of the fly. So when we're looking at this fly, thoracic segment number 1 is kind of tucked away up here. We can't really see it very well. This is thoracic segment number 2, on which the wings develop. And thoracic segment number 3 is right here, and it doesn't usually develop wings. Instead, what it develops are these little balancing organs called halteres. That's normal development. Now, the reason that this third thoracic segment develops the way that it does is because it expresses this ultrabithorax homeotic gene, which represses all of the genes that would otherwise lead to the formation of this anatomical structure that we call T2, right? And the wings and bristles associated with it. So if we knock out ultrabithorax expression in T3, there's nothing telling this thoracic segment number 3 that it's not thoracic segment number 2. And so what we see happening is that we see a reiteration or repetition of thoracic segment number 2, and we end up with a fly that has 2 thoracic segments number 2 and no thoracic segment number 3, right? So loss of expression of an important homeotic regulator of the formation of body structures leads to disruption of the body structures that are formed, or what's called a homeotic transformation. Another example of a homeotic gene that provides pretty dramatic effects when we misexpress it is antennapedia. Antennapedia is a transcription factor that's switched on, and usually it's switched on in the specific regions of the 3 thoracic segments where we're going to see legs forming. So when antennapedia gets switched on in T1, T2, and T3, at those positions we see genes being activated that lead to the formation of legs in those regions. Well, what if we take that antennapedia gene and instead of expressing it in the thoracic segment, now we express it in the head where it's not usually expressed. What we see then is that in the region where these little feathery arista-like antennae form, instead we see legs forming because of the ectopic expression of antennapedia in a region of the developing fly where it's not usually expressed, right? Wherever antennapedia gets expressed, that switches on the set of genes that make leg. And so we see leg being made. Conversely, a gene that's required for the formation of eye, called iris, it's normally expressed in the region of the head where the eyes form, and it's critical for that formation. If we express that gene in other regions of the fly, then what we see is a phenotype where the fly shows extra eyes. Anywhere that eyeless gene was expressed, we now see genes being switched on that attempt to lead to the animal's anatomical structure, the eye, right? They're not normalized, they don't have neuronal connections, they don't work, the fly doesn't see through them, but you can definitely see these structures forming on other regions, including the legs and along the back, right? So key thing to recognize here is that when these homeotic genes get switched on, downstream of those segmentation genes, they actually flip the switches on the genetic programs that lead to specific body structures forming. Now, if we look at the fly genome, what we would see is a cluster of homeotic genes that are present in a couple of different complexes. These are related to one another. They're a family of genes, and each homeotic gene is responsible for a particular body structure specification, right? And so that's what we see in Drosophila. Now, are these limited to Drosophila? Oh no. Hox genes are found actually in all animals in which we've looked. And their importance in development differs depending on which phyla you're in, but definitely in all bilaterally symmetrical animals, they're critical for development, highly, highly conserved across phyla. So what we see in different species is that there's a whole family of these homeotic proteins, these transcription factors that turn on the genes that specify body structures. And these are often referred to as Hox genes, right? These homeotic genes that can cause homeotic transformations are Hox genes, and Hox stands for homeobox. And the reason that they're called homeobox genes is because the transcription factors that they encode have a conserved DNA binding domain. In other words, if you line up the sequences of these proteins that are encoded by the Hox genes across all these different organisms, you see that they have this about 180 nucleotide long DNA sequence that specifies a domain on the protein that binds to DNA, and these are called homeodomains. So that's what makes them so similar in terms of the function that they're carrying out. Now, as we've talked about before, anytime we see this kind of a gene family or superfamily, we know that what's happened is that we've seen gene duplication events over evolutionary time, right? We can see non-reciprocal recombination events leading to gene duplication. We can see translocation events that lead to representation of these genes at different locations in the genome, and both of those seem to have happened many times over evolutionary time, which has expanded the Hox gene family tremendously. So here's the idea. You know, originally in some primitive ancestor, we had a Hox-like gene, only one copy. Duplication event, we now have 2. Another duplication event, we've now got 4. And so in our, early bilateral ancestor, we had a cluster of Hox genes that may have looked something like this, 7 Hox genes, right? Even in jellyfish, and we look at invertebrates like jellyfish, we can see that they have relatives of the Hox genes. They've lost some, but we can still detect that there are some genes present that have sequence similarity to these Hox genes that we find in other animals. If we look across phyla, we see if we come down here to arthropods, insects like Drosophila, we see that cluster that we just saw on the previous slide. If we look in tetrapods, vertebrates like us, human or mouse, what we see is not one set of Hox genes, but actually that those Hox genes have been duplicated and translocated so that they're now actually on 4 distinct chromosomes. So we have 4 sets of these Hox genes, but the sequence similarity is you know, leaves no doubt as to the evolutionary relationship between these clusters and sets of Hox genes that we have distributed across 4 chromosomes, and the ancestral Hox cluster that is the evolutionary ancestor to those found in all these other animal phyla. And another you know, bit of interesting genetic consequence of this evolutionary relationship is that these genes can functionally compensate for one another. If you have an organism that has lost the function of an antennapedia gene, you can actually take the sequence of the antennapedia gene from another species and put it into that organism, and it will compensate. It'll be used exactly as the endogenous Hox gene would be. So in other words, you can make a mutation in fly that knocks out antennapedia, take antennapedia from mouse and use genetic tricks to get it into the fly, and it will take the place of the Drosophila antennapedia gene. So just really indicating the evolutionary conservation, not only in sequence but also protein function, that we see in these highly related genes. So because we have multiple copies and duplicate sets of these homeotic genes, these homeobox genes, our developmental program is actually quite complex compared to these other animals, as you would imagine, right? And what we see during our developmental program is that we have the interaction between multiple homeotic genes that are specifying these effector genes being turned on and formation of body structures. So we don't typically see the same dramatic homeotic transformations in human that we see, for example, in fly. If we knock out one antennapedia gene, or we see misexpression of one antennapedia gene, for example, in human, you know, those genes are associated with limb development in human. They're not called antennapedia, they're referred to by number, but we're not going to see a leg growing out of somebody's head, in other words, right? So we don't see those dramatic effects because we have redundancy and multiple interactions with different Hox genes with one another to specify a developmental program. So we see more subtle developmental effects typically, right? Not entire missing limbs or other anatomical structures, but alterations in the developmental program, so they're not quite normal. One example of that, one of the more common examples of that, is syndactyly or polydactyly. Syndactyly refers to the fusion of digits in the, you know, hands or feet. And polydactyly refers to the presence of extra digits. Well, these digit formation is very well characterized to be regulated by Hox genes. And one specific Hox gene in human, Hox D13, if you have a mutation in that Hox gene, that leads to the clinical presentation of synpolydactyly, where you have both fusion of digits as well as extra digits being formed, or parts of digits being formed. You can see extra toes, extra fingers. And interestingly, other Hox gene mutations have been identified in human genetic disorders, like disorders that affect nervous system development, either central nervous system development or peripheral nervous system development. And there are, for example, peripheral neuropathies that are associated with Hox gene mutation. Interestingly, defects in Hox genes have also been implicated in susceptibility to particular cancers, most notably breast and prostate cancers. So they are, you know, very relevant to human physiology and control important aspects of our anatomy and development, just as they do in these other organisms, all the way back to fruit fly. And you know, it cannot be overstated how much of a surprise it was to the biological world when the details of development were worked out in a model organism like fruit fly, which I don't think anybody would be able to mistake for a human. But then we discovered that extremely similar genes and gene programs were being initiated in us, in mouse. It was just a huge shock. Nobody expected that our development would be so highly conserved, given how different our anatomy and physiology are. All right, so we talked about a lot of stuff there. I want to just go back a bit and kind of summarize conceptually what we just saw happening in Drosophila development, and sort of tie it all together. So remember that we started with a gradient of these morphogens, bicoid and nanos, such that at the region of the embryo that's fated to become anterior, and remember this was even before we had fertilization, so really in the oocyte, we had a high concentration of bicoid protein because bicoid mRNA had been specifically localized to that region of the oocyte. At the region of the embryo that's going to become posterior, we had a high concentration of nanos protein for the same reason, right? Nanos mRNA was localized there. And what we saw was a gradient of expression, right? As this bicoid protein sort of diffuses away from the site of localization, we get this gradient of expression where it's highest here at the anterior end, lowest or non-existent here at the posterior end, and the converse for nanos. And so any position along this AP axis is getting information in molecular form from these morphogens as to where it is in the embryo. So if we're here in the embryo, what are we seeing? We're seeing high bicoid, right, and low nanos, or no nanos at this end. And so let's imagine that these proteins, bicoid and nanos, remember they're transcription factors, they go on to interact with, let's say, just genes A, B, or some set of these, gene C. We've got three downstream genes that are going to be responsive to these morphogen gradients. And when you have high bicoid, low nanos, let's say that we see gene A specifically being activated. Genes B and C stay off, right? And so because we've activated gene A, maybe gene A encodes transcription factor. And so now we express in these cells at this region of the embryo transcription factor A. Well, what is that transcription factor then going to be doing? Well, it's going to be, in turn, regulating the expression of other genes. So let's say downstream of transcription factor A, we switch on genes D, E, and F, right? And downstream of those genes, which are also transcription factors, we switch on additional target genes, right? So we now have transcription factor D that's hitting specific genetic targets in the genome. We have transcription factor E that's hitting specific targets in the genome. We have transcription factor F that's hitting specific targets in the genome. And so we get target gene regulation taking place downstream of this genetic program that is going to lead to finer and finer specification of what the cells in this region of the embryo are destined to become. So we initiate a morphogen gradient which switches on a gene, right? This is a very simplistic example. Obviously it'd be more than one gene, which leads to expression of a specific transcription factor, which switches on target genes, which may encode additional transcription factors, which are going to switch on additional target genes, and so on and so on. So we get this cascade of regulated transcription that leads to these very finely regulated genetic programs that specify the cell fate of the cells that are in this region of the embryo. Here at the other end, what would we be seeing? We'd be seeing low bicoid and high nanos, so the opposite scenario. And same genes in the genome, right? These cells have exactly the same genes, but now because we've got high nanos and low bicoid, we're not going to see gene A switched on. Gene A is going to stay off, and maybe we see gene B switched off as well, or stays off, and instead gene C is switched on. And because gene C also encodes a transcription factor, we're going to see a different set of genes switched on. So now genes G, H, and I, for example, right? Here in the middle region, what are we going to see? Bicoid approximately equal to nanos, right? Now let's say, same genes, gene A, gene B, and gene C. But now let's say that gene B is specifically switched on, genes A and C stay off in response to this. And so now we get transcription factor be expressed, right? Which switches on downstream genes K and L, whatever they do, right? Key point here is that these morphogens that are initially expressed in the oocyte switch on different sets of genes that encode different transcription factors, which are themselves then going to switch on different sets of genes, which may encode transcription factors that switch on additional sets of genes. Ultimately, those target genes at the end of this cascade are going to be genes that actually define the actual identity of these cells, right? So we get this finer and finer specification of cell identity that initiated with these morphogens and their concentrations within the embryo, right? So we get initially a cell that has received information that tells it you're anterior, right? So anything in this region is going to have that very general cell fate specification. But as a result of different genes that get switched on, now we're going to get a little bit of a finer specification. So you're not just anterior, you're head, or you're thoracic 1, or you're thoracic 2, or you're thoracic 3. So still anterior, but now your fate has been defined a little bit more specifically, right? Additional genes get switched on. Now, you're not just head. Maybe you're specifically going to become eye. You're specifically going to become antennae. You're specifically going to become mouth parts. So your identity and fate is becoming more and more specifically defined with every additional set of genes that get switched on. Now you're not just an eye, you're going to be a specific neuron in the eye. You're going to be providing structural support. You're going to be an enzyme that produces pigment, right? So specific genes that are required to form the anatomy and lead to the physiology of the eye, and so on and so on and so on down the line, right? Getting more and more specific as we go. So this initial specification, very general, was initiated by our maternal effect genes. And then we got those segmentation genes that got switched on, that defined more precisely what anterior and posterior meant all along the anterior-posterior axis. And from then we get these homeotic or homeobox genes that actually lead to effectors being turned on that lead to specific body structures, right? So that's what we've been talking about. But the same sorts of morphogenic cues that we see happening in Drosophila development, they're also regulating vertebrate development. If we look in mouse development, we see, not bicoid nanos, but other related morphogens providing this kind of positional information as well. And so you can have, let's say in early embryonic development, you've got a ball of cells that are all fated to become some part of the embryo. And now this cell starts to produce a morphogen, right, some chemical signal, and secrete it out of the cell. Well, that signal is now going to diffuse away and is going to be signaling to the other cells in the embryo in a concentration-dependent manner in a very similar fashion as to what we see in Drosophila. As I said before, there are more things in common in our developmental program than things that are different if we look at the molecular level and how this actually plays out. So again, this was, 2 examples of different mechanisms of regulating gene expression, the expression of a gene's product, what's produced, where it is in the cell, and that actually initiated a whole host of transcription factor signaling that led to specification of the embryonic body plan. Pretty much any developmental process that we could take a look at is going to be governed by very similar mechanisms, as well as if we think about those other mechanisms that are possible, mRNA degradation, alternative splicing, microRNA regulation, all these other forms of gene regulation also come into play.